Welcome to Unit 7 for Honors Biology. In this unit, we will talk specifically about protein synthesis. That'll be our focus for this unit. Now, along the way, I have told you that we have to do several different things before we get to the coveted unit of genetics. This is the last part of the puzzle that you gotta know in order to go on to genetics. So let's do a recap of what we know. First thing you know is how DNA replicates itself. You know the process of DNA replication. You know how I can take one strand and make two strands of it that will look identical to each other. Second thing you know is what I sometimes use DNA replication for, and that is mitosis or meiosis, depending on what cell it is I'm talking about. You know these processes. So the last piece of the puzzle that you now have to go into is how our DNA sends out its information that it has. How does it get the information that makes you, you, and me, out of its genetic code, that ATCG? How does it go to something else that our cells can use? And that, of course, is going to be the very last thing, the very last piece of the puzzle, and that is that protein synthesis. With protein synthesis, I'll go ahead and let you know that it can be broken down into two steps. Your first step is known as transcription. This is where I take my DNA and I turn it into RNA. And we talked a little bit about RNA when we were doing our DNA resume. DNA and RNA are best friends. But this is how I get the information out. I take my DNA strand and I turn it into an RNA strand. And so we'll get into what that actually means. The second step, once you transcribe it, you now have to translate your RNA strand. So for RNA, once we get it to that stage, I then take my RNA strand that I just created and I turn it into what we call a protein, which at its core is going to be made up of amino acids, the building blocks of a protein. So first off, with protein synthesis, when we were talking about DNA replication, I asked you, where does DNA replication happen? Well, it's got to happen in my nucleus. My DNA is too valuable that I cannot let it go outside of its nice little protective bubble that is the nucleus. So if I want to get my information from my DNA out, I have to have a go-between. I have to have a mediator that can take it from the nucleus, get that information, transcribe it from DNA, and then take it out to my ribosome, which is where my proteins are going to be made. And that is the basis of protein synthesis. Now, to start with, you first have to understand RNA. Now, I introduced RNA when we were talking about DNA. This is DNA's best buddy. They are just a little bit different. So as a recap, just to go back over the stuff you already know, RNA is ribonucleic acid. From this, you can already tell I have one big difference between RNA and DNA. So one of my big differences is that I have ribose instead of deoxyribose. My sugar has changed. One of the other differences that we talked about is that it is single-stranded. Since I'm transcribing it, I don't need RNA to be double-stranded. I am only interested about getting the information out of DNA and taking it to my protein. And a single strand is way easier to work with than a double strand. So I just have a single strand of RNA that will come out. My third difference is that I have uracil instead of thymine. So one of my nitrogen bases has been swapped out. DNA is always C, G, A, and T. With RNA, it's C, G, A, and U. So uracil comes into play. So when you transcribe DNA to RNA, you've got to remember thymine no longer plays a part. If you're talking about RNA, you got to use uracil. Then the last one that I want you to know is that RNA is usually only one gene long. My DNA in every single one of my cells is the same. No matter what, my DNA, the DNA that is going to be held in that nucleus is going to be the same whether I am a lung cell, stomach cell, brain cell, whatever. But we all know that brain cells are different than stomach cells. Stomach cells are different than lung cells. What I actually pull out of my DNA is what is important to that cell, for that body part, for that body system. So I don't need all of my DNA. I don't need all of that information, because some of that information that's stored there doesn't pertain to me as a lung cell. It's for the liver, not the lung. So I don't need to transcribe that. With protein synthesis, it's very unique for each cell that you're talking about, but it occurs the exact same way no matter what. But I'm only going to take the information that I need. So I'm only going to look at usually just one gene at a time and just take that information and then move on. Now, just to go through my differences just a little bit more and get more familiar with RNA, let's talk about what's in an RNA nucleotide. Still the building block, just like it was in DNA. You're still going to call this a nucleotide, but it is going to have some subtle differences with it. One thing is you're going to have a ribose as your sugar instead of deoxyribose. So you're going to have ribose coming in. You'll still have a phosphate group, and you'll still have nitrogenous bases. So your bits are still roughly the same. You just have minor differences in that. With your nitrogenous base, you're now dealing with A, U, C, and G. You no longer have T involved. Whereas phosphate doesn't change at all. That's the only one that stays exactly the same between DNA and RNA. In this picture that I'm showing you right now is the difference between ribose and deoxyribose. Really, the only thing that is different is deoxyribose is missing an oxygen, whereas ribose has it. In this next picture, you're looking at uracil versus thymine. Uracil, it's taking the place of thymine, so I'm still going to be two rings. So I'm still going to fall into the same category of the different types of nitrogenous bases. But looking at it, you'll notice that there are subtle differences between the two, and that is unique for what RNA needs to do. Just to kind of reiterate it, C still equals G, but now when you come across an A, you got to pair it up with U. T no longer exists, so you cannot use T anymore. So let's go ahead and just practice that. Say I gave you this strand of DNA. T, G, C, A, T, C, G, T, T, A, G, and then C. If I was coming up with my complementary RNA strand, so I've got to pair everything back up. If I come across a T in DNA, I automatically will pair it with A. Then my G would of course pair with a C, C pairs with G. Now I'm at an A. T no longer exists, so A now has to pair with U. Then I'd have an A, G, C, A, U, C, G. So it works the exact same way. It's just you have to remember that anytime you come across an A in the DNA, it now has to pair up with U in the RNA. Now the other interesting bit about RNA is I have a couple of different types of RNA. I actually have 3 different types, and each one is specifically tailored for the job that it has to perform within the process of protein synthesis. So I will start with the very first RNA that you will come across when you are going through the process of transcription. And that very first one is called mRNA. This stands for messenger RNA. So think of it like a bike messenger. Bike messenger comes, say he's working for Jimmy John's. He comes and he picks up the sandwiches and then brings them out to wherever it is you are. And from the commercials, it's freaky fast. With this, I'm getting my information from DNA and I'm taking it out to the ribosome. So for this one, it is the messenger between DNA and the ribosome, which is where protein synthesis occurs. What this does is it carries the copy of DNA to a ribosome. On the picture that I am now showing you, you have your DNA double helix. It gets split just like it normally would for DNA replication, and I will have a polymerase, it's called RNA polymerase, come in and it will make a strand of mRNA that can then, once it's done and it's got the information that it needs, it's made the copy that it needs, it will detach itself from the DNA double helix. The helix will reseal itself, and the mRNA strand is now free to leave the nucleus, and it'll go out into the cytoplasm and it'll find a ribosome and start going through translation. The second type of RNA that you will come across is rRNA. This stands for ribosomal RNA. What these are, it's one of many proteins that make up the ribosome that you guys are familiar from all of our cell part stuff that we have done. Ribosomes are important because that is the site of protein synthesis. That is where proteins are made. So this is a pretty important part of the process. In the picture that I am showing you, you have two different types of rRNA that are involved. You have one that helps make up the large subunit of a ribosome, and you have another one that makes up the small unit of a ribosome. But everybody just generally refers to them as rRNA, and you're good to go. The very last RNA that you will come into contact is tRNA. This is known as transfer RNA. What this one does is it transfers, keep in that kind of mode, amino acids. And in most of my notes, I will now start to refer to as amino acids as just AA. It's usually just how I abbreviate it. So you'll start to see that more and more. So just be aware that when I say AA, I do mean amino acids. But it transfers amino acids to the ribosome so that these amino acids can eventually be hooked together and form the protein that I am interested in. In the picture that I'm showing you, tRNA looks a lot like a T. It has its own unique genetic makeup that will break down a little bit so that you know how it kind of works. I'll start throwing in words such as codon and anticodon when we talk about tRNA. This one is pretty important because it does bring in those amino acids so that I can build my protein chain. And eventually that's what I'm going to be mostly interested in. For now, I am going to stop this lecture, go and do the other homework assignment that I assigned to you, and then I will see you in class.